Hello everyone, so today's video is on Alzheimer's disease. We're going to be covering the neuropathophysiology behind Alzheimer's disease. We're looking at the proteinopathies behind Alzheimer's disease, as well as the immunological components behind Alzheimer's. And in the second half of this video, we're going to be looking at pharmacological interventions, the mechanisms behind therapies, how they either suppress symptoms or reduce pathology load, plaque load. And we're going to be looking at novel or upcoming treatments as well. Now, medically speaking, Alzheimer's disease is characterized as a progressive, irreversible neurological disorder of the brain. And symptomatically speaking, it's associated with a drastic loss in cognition. And in later phases of the disease, when you see significant neuronal loss, one's ability to carry out everyday tasks is completely diminished. Unfortunately, there is no cure for Alzheimer's disease. What there is available are pharmacological interventions that aim to suppress symptoms and also reduce disease load or plaque pathology, which we'll get into later. Now, Alzheimer's itself is attributed to an abnormal buildup of two distinct proteins, and these proteins act as molecular hallmarks in diagnosis. So medics will look out for these in brain scanning techniques to kind of distinguish Alzheimer's from other progressive neurological conditions. So, for example, multiple sclerosis is an example of another neurological condition, and the molecular hallmark of multiple sclerosis is a drastic loss in that myelin coat around the axons of neurons. So that demyelination acts as that kind of molecular hallmark, and it allows the medics to kind of make a distinction and a decision on what's wrong with the patient. And Alzheimer's is no different. Those molecular hallmarks that they look out for especially is that build up in extracellular amyloid beta plaques and that intracellular buildup of neurofibrillary tangles. Now, those neurofibrillary tangles are composed of hyperphosphorylated tau protein. Now, these proteinopathies essentially synergize and they amplify downstream toxicity. They encourage a pro-inflammatory neuroimmunological response, and that pro-inflammatory response encourages further pathological processing of these proteins. So you get an exacerbated buildup, and then that exacerbated buildup will then amplify this kind of pro-inflammatory response again, and that pro-inflammatory response will encourage further pathological processes. So it's almost like an endless loop, and that kind of speaks to its progressive nature, unfortunately, which we're going to get into as we kind of move through these slides. So in order to really understand the pathology behind Alzheimer's disease, I think it's really important to kind of start from the beginning, and we're going to be looking at amyloidogenesis first and the formation of these extracellular amyloid beta plaques. And those amyloid beta plaques kind of govern downstream tau hyperphosphorylation. So the plaque buildup is seen in very kind of early phases of the pathology behind Alzheimer's, and that kind of acts as a catalyst and a trigger for the rest of the steps that follow downstream. Now, both proteins, APP and the intracellular tau protein and the APP protein that's expressed in our transmembrane in our neurons, they play really important roles. Under normal physiologic conditions, they play important roles that favour neurite outgrowth, synaptic plasticity, synaptogenesis, those kind of processes that kind of speak for our central nervous system, our ability to learn, our ability to think, cognition, our ability to relearn things. But kind of just to get into a bit of background, the amyloid precursor protein itself is the transmembrane glycoprotein that's expressed particularly in high densities in the synapse, but also in the kind of axonal membranes of some neurons as well. And it's a glycoprotein, and when it's cleaved by alpha secretase, and that cleavage generates this kind of soluble alpha APP fragment, and that acts as a neurotrophic fragment that promotes neurite outgrowth. It works along with the glutamatergic system to enhance long-term potentiation and the maintenance of synapses. But its pathological processing is through the enzyme beta secretase, and when beta secretase kind of degrades that transmembrane protein, you get these kind of toxic peptides that will kind of aggregate and oligomerize to form the amyloid beta plaques, and those amyloid beta plaques kind of trigger our immune system, and it's that kind of pro-inflammatory response that's then thought to kind of initiate tau hyperphosphorylation. So it's really important to kind of make to kind of make that connection, when understanding the pathophysiology of Alzheimer's, that kind of connection between the generation of amyloid beta aggregates and oligomers in the extracellular matrix and the downstream effects it has on tau hyperphosphorylation and neuroimmunological kind of inflammation. So I've got a diagram here displaying the amyloid precursor glycoprotein and where it kind of sits within our neurons. Now that is kind of sitting in that lipid bilayer, that phospholipid bilayer, that membrane of our neurons, and it has a extracellular domain, which is where the AB amyloid beta domain is, and that's where either our alpha secretase or beta secretase will cleave, and it cleaves at different, depending on the enzyme, will cleave at different portions of that domain, and that is dependent on either the amyloidogenic or the pathogenic pathway or the normal physiologic pathway, which is favored by alpha secretase. Now you have a transmembrane domain, and that's the hydrophobic or the lipid soluble domain. And then you have the C-terminus domain, which is on the intracellular part of the neuron. And that transmembrane and intracellular domain is cleaved by gamma secretase in both the amyloidogenic and the physiologic pathways. Now, alpha secretase will cleave that within the AB domain, and beta secretase will cleave at the N-terminus of the AB domain, kind of keeping it intact, which is guaranteed to form those amyloid beta oligomers. Alpha secretase activity, because it forms that soluble APP alpha fragment, because it cleaves within the AB domain, there's no chance of forming those amyloid beta peptides. Now, essentially what's happening with the APP membrane glycoprotein before disease or pathology in Alzheimer's is initiated is there's an increased level of APP trafficking into the cell, so APP endosomal trafficking into the cell. So that phospholipid bilayer will fold in on itself and form these small endosomes, if you like, similar to vesicles or the same as vesicles. And this is usually under stress, so exposure to harmful stimuli, pro-inflammatory stimuli, whether that's through diet, lifestyle, and there's an increased number of APP trafficking, and those APP endosomes are more acidic in their pH, so that acidity favors beta secretase activity, and obviously beta secretase will then cleave at the N-terminus of that AB domain, and it will give you your peptides that will, those amyloid beta oligomer peptides that will be ready to aggregate and form these plaques. So I've got a table here and a diagram to kind of help people understand the differences between the physiological processing of the amyloid precursor glycoprotein compared to the pathological processing of APP. Now, the pathological processing is referred to as amyloidogenesis because that pathway, that amyloidogenic pathway, will result in the formation of those amyloid beta peptides that will eventually oligomerize, aggregate to form those senile plaques you see in Alzheimer's disease. Now, I'll start off with the physiological processing of APP. Now, the physiological processing of APP is initiated by the enzyme alpha secretase, and that enzyme is coded by the ADAM10 gene. So typically in normal physiological conditions, ADAM10 gene expression is upregulated. Now, alpha secretase cleaves within the amyloid beta region of the APP protein, and that's a really key step in preventing the formation of those amyloid beta peptides. Now, when alpha secretase cleaves the APP within the AB region, you get the generation of what's called a soluble amyloid precursor protein alpha fragment, and that's the neuroprotective synaptogenic or neurotrophic fragment that participates in extracellular signaling mechanisms that will favor or facilitate neurite outgrowth, synaptogenesis. So it's a neuroprotective protein fragment. And what's left after alpha secretase cleaves that APP or that portion of the APP protein is a C8 free fragment. That's the kind of membrane bound portion of the APP protein, the lipid soluble hydrophobic portion that's sitting in the transmembrane of the nerve cell. And that C8 free fragment is cleaved by gamma secretase to form a non-toxic P3 peptide and the APP intracellular domain, or the AICD. And both the P3 peptide and the AICD will participate in intracellular signaling mechanisms, mainly facilitating gene regulation of gene expression, but it all kind of contributes towards neuronal or neural protective mechanisms. Now, when we look at the amyloidogenic pathway, beta secretase initiates that kind of cleavage instead of alpha secretase, and beta secretase is coded by the gene BACE1. Now, I just want to make it clear that BACE1 gene expression is upregulated in later phases of Alzheimer's when you have that kind of pro-inflammatory environment, and I'll get into that in the coming slides. But when amyloidogenesis, before amyloidogenesis is kind of initiated, before we get that buildup of plaque, there's not necessarily an upregulation in BACE1 expression. But there is as I mentioned before in the previous slide, there is an increase in the amount of cell-mediated endocytosis, or APP trafficking, endosomal trafficking. So that APP proteins being trafficked into the cell in these endosomes, and that kind of changes the intracellular environment, and it favors beta secretase's catalytic efficiency. Those endosomes are slightly more acidic, okay, and that creates a more acidic intracellular environment, and beta secretase just works more optimum at that environment. Its catalytic efficiency is favored, and alpha secretase doesn't. So beta secretase is essentially outcompeting alpha secretase, okay. So that kind of overactivity of beta secretase initially will result in the formation of those kind of senile plaques, or those amyloid beta peptides, which will eventually become those senile plaques. Anyway, to take a few steps back, beta secretase will cleave at the N-terminus of the amyloid beta region on the APP protein, and that is the really important step in generating the amyloid beta peptides, okay, particularly the amyloid beta 42 species. Now, when beta secretase cleaves at the N-terminus of the amyloid beta region, what you get left is a membrane-bound or membrane-anchored C99 fragment. Okay, now that C99 fragment contains the full amyloid beta sequence intact. So when gamma secretase comes and cleaves that, you get the generation of those amyloid beta peptides, either the amyloid beta 40 species or the amyloid beta 42 species, which are more hydrophobic, and they're more aggregation prone. They're more prone to oligomerizing, eventually aggregating and forming those plaques. And you also get the generation of the APP intracellular domain. So you get the generation of the APP intracellular domain in both the physiological and the pathological processing of the APP protein. Now, that C99 fragment is different from the C83 fragment. Both fragments are membrane-bound hydrophobic fragments, but because the initial enzyme, because beta secretase is cleaved at the N-terminus, okay, of the amyloid beta region, it's left that amyloid beta sequencing still intact in that C99 fragment, which means that when gamma secretase comes and cleaves, it will form those amyloid beta peptides, whereas in the physiological pathway, alpha secretase has cleaved it within the amyloid beta region, disrupting that sequencing altogether, leaving that kind of membrane-bound C83 fragment, which does not contain that full sequence. So when you get that cleavage from gamma secretase, you won't get your amyloid beta 40 or 42 species being generated. Now, just to kind of once we kind of get those amyloid beta peptides, especially those amyloid beta 42 species, when we get sufficient amount of oligomerization and aggregation, we start to get the presence of these plaques. The plaques start building up, and our microglia and our astrocytes will come, particularly the microglia. They express these kind of TREM2 receptors, these toll-like receptors, which will kind of see the presence of those plaques as a form of DAMP, or a damage-associated molecular pattern. Now, this kind of initiates inflammasome signaling, or that kind of the activation of that NLPR3 inflammasome complex, that intracellular complex, that will upregulate the expression or gene expression of pro-inflammatory cytokines in microglia. So they're going to become activated, okay, and they're in turn going to activate astrocytes. The astrocytes will go into astrogliosis, and then this kind of creates a hostile inflammatory environment. And all these pro-inflammatory cytokines that are being secreted, like tumor necrosis factor alpha, like interleukin beta, interferon gamma, they're going to change the neuron. They're going to change the neuron on a transcriptional level. They're going to cause transcriptional shifts that will result in the upregulation of BACE1 expression, okay, and the suppression of ADAM10 gene expression. So when BACE1 expression is upregulated, you're going to get a favor in amyloidogenesis. It's going to favor the amyloidogenic pathway, okay, and ADAM10 gene expression will be downregulated, so you'll get less physiological processing of APP, okay. You'll get less, and as you know, the physiological processing of APP is really important in generating those neuroprotective protein signalers that will kind of favor synaptogenesis and neural outgrowth, as opposed to the pathological processing, which does the complete opposite. Okay, these plaques will result in this kind of inflammatory response, which will kind of initiate this this disease. Now, a consequence of microglial activation and astrocyte activation, because of the presence of these DAMPs or these senile plaques, okay, is downstream tau hyperphosphorylation. It's suppression in base 1 expression, but it also disrupts or dysregulates calcium homeostasis, which will activate enzymes in like caspases and calpains, lowering the threshold for apoptosis and neuronal cell death. It also results in excitotoxicity, because reactive astrocytes, they're kind of compromising their glutamate uptake, so you get prolonged glutamatergic signaling on neurons, which will result in an intracellular influx of calcium, as I said, a dysregulation in calcium homeostasis within the neuron that will kind of lower that threshold for apoptosis. So you can kind of see how this kind of amyloidogenesis has initiated has kind of initiated this positive feedback loop, this over amplification loop of activity, this kind of pro-inflammatory assault, and then the further formation of plaques, okay, and the further hyperphosphorylation of tau protein, which I'm going to talk about more as we kind of progress throughout this video. So the cleavage of or the breakdown of our amyloid precursor protein isn't necessarily a spontaneous process. It just happens spontaneously. There are reasons why why the beta secretase or amyloidogenic pathway is favored in the first place over our normal physiologic processing of APP by alpha secretase. And one of the reasons is an increased amount of APP endocytosis or cell-mediated endocytosis. So as it sits, that amyloid precursor protein or that glycoprotein is sitting in the transmembrane, that lipid bilayer of our neurons. And during endocytosis, as most people will be aware, that kind of transmembrane or that phospholipid bilayer will partially kind of fold in on itself, and a portion or a small portion of that lipid bilayer will become a kind of like an endosome with the APP protein inside of it. And an increased endosomal trafficking leads to a more kind of acidic environment. Now, that acidic environment favors beta secretase's catalytic efficiency. It's more efficient as an enzyme or as a catalyst at that pH. Alpha secretase isn't. So it's going to beta secretase is going to be almost out competing alpha secretase, and you're going to get that amyloidogenic breakdown. And as you know, beta secretase cleaves at the N terminus, and that C99 fragment still has that amyloid beta sequencing intact. So when gamma secretase will come and cleave that, you'll get that amyloid beta 40 or 42 species. Okay, and that 42 species is really prone to oligomerization. And furthermore, Alzheimer's isn't just like this kind of random build up of proteins. It's a very regulated, stress driven process, okay, and it involves a shift in neuronal processing on a transcriptional level, and it also causes a shift in microglia and astrocyte activity as well. So what's happening here is, once we've got an upregulation in beta secretase activity and increased amount of kind of endosomal APP trafficking, you're going to get more of a build-up of those amyloid beta aggregates. And our microglia, as I said before, they contain these kind of like toll-like receptors, and once they become activated and their inflammasome signaling is switched on, there's going to be an upregulation in the expression of pro-inflammatory cytokines like tumor necrosis factor alpha, like interleukin 6, interferon gamma, interleukin 1 beta. And those pro-inflammatory cytokines will directly bind to the neuron, for example, like tumor necrosis factor alpha in particular, and it will cause a transcriptional shift in neuronal processing. So as I said before, under normal physiological conditions, ADAM10 was upregulated. Its expression, the ADAM10 gene, which codes for our alpha secretase, which favors our physiologic processing of APP, was upregulated. So you're getting more appropriate cleavage of the APP precursor protein, which is what we want. But because of this stress-driven response for our microglia through beta secretase activity, we're now favoring base 1 expression. Okay, so tumor necrosis factor alpha, when it binds to the neuron, it initiates a cascade of intracellular signaling proteins, and it eventually results in the activation of, say, transcription factors like NFKB, and that will directly suppress ADAM10 expression but favor or upregulate beta secretase expression, so base 1 gene expression. So now we have an increase in beta secretase activity now in our inflammatory state. So you're going to get more of that cleavage, and furthermore, you get what's called cytokine-mediated endocytosis. So our neurons are actually going to be taking in more of those proteins. So you're going to get more endosomal trafficking. Okay, so everything that's happening now is favouring beta secretase and not favouring alpha secretase. So we're actually favouring the amyloidogenic pathway, and this is essentially a amplification loop, like you see with most pro-inflammatory diseases, whereby you have your damage-associated molecular pattern, your immune cells, this could either be in your periphery, but in this case we're talking about our central nervous system. Your immune cells, like your microglia and astrocytes, are going to detect those damage-associated molecular patterns, and they're going to activate and shift into a pro-inflammatory state, and they're going to secrete pro-inflammatory cytokines, which is going to encourage or keep the neuron in that transcriptional shift, that pro-inflammatory shift in that state. So it's going to be upregulating enzymes like beta secretase, which are then going to be cleaving that APP at an abnormal rate, and you're going to get more of those amyloid beta 42 or 40 species. You're going to get more aggregation, more oligomerization, and more plaque buildup. So it's essentially this kind of positive feedback loop that's working round on itself, and that's one of the reasons why it's so progressive in nature, because those plaques are going to get bigger, and obviously we know that those plaque formation governs downstream tau hyperphosphorylation, because a lot of those pro-inflammatory cytokines will favour the activation of certain enzymes in neurons like p38-MAP kinase, which is very associated with tau hyperphosphorylation and the formation of those neurofibrillary tangles. And it's just the molecular hallmarks are going to get bigger and bigger and bigger, and our astrocytes and our microglia are going to get more and more agitated, if you like, and they're going to keep secreting those pro-inflammatory cytokines, and they're also going to activate each other. So the microglia will activate the astrocytes, and the astrocytes will go into astrogliosis. They'll secrete their reactive oxygen species and pro-inflammatory cytokines, and it's this endless loop of inflammation and activation, and it kind of keeps the neurons in that kind of amyloidogenic state, if you like. That transcriptional shift almost becomes permanent. So that downregulation of ADAM10 and upregulation of BACE1, and you have a downregulation of phosphatase 2A enzyme and an upregulation of kinase enzymes, and all of this favours amyloidogenesis and tau hyperphosphorylation. So before we kind of get into the neuroimmunological mechanisms behind Alzheimer's disease, I just want to quickly touch on risk factors, or particularly genetic risk factors in Alzheimer's disease. Now, some of the risk factors that I've not mentioned on this slide are age, so older people are more likely to develop Alzheimer's, people who smoke, people who eat, pro-inflammatory foods. There's many alcohol, drugs. There's many risk factors to Alzheimer's disease, but a lot of it is to do with whether you're genetically predisposed. There are certain alleles within the population that are more correlated or very associated actually with the development of the disease later in life, especially when coupled with a high inflammatory lifestyle, which I think most people in the 21st century are kind of guilty of, because it's the only option. It's what we're given, our food. A lot of our food is processed. Even healthy food now is not necessarily as healthy as we probably think it is. And and when that coupled with this kind of like genetic vulnerability can leave you a lot more likely to develop or manifest Alzheimer's later on in life. Now, there's a subset of genes called the APOE genes, and that codes for a protein which eventually, after post-translational modification, becomes a lipoprotein. So it's referred to as a lipoprotein. Okay. An A-lipoprotein E, and it's a lipid transport protein, and it's associated with cholesterol trafficking, synaptic maintenance, neuronal repair, and also it's expressed quite heavily in astrocytes and microglia, and it's involved in say glial-mediated phagocytosis as well. Now, there are three isoforms, or there are three alleles of this gene within the population, and the first two, the APOE2 and the APO3, isoforms of the A-lipoprotein E, aren't considered a risk factor for Alzheimer's disease. It's the APOE4, so the A-lipoprotein E4 allele, that's the strongest genetic risk factor for Alzheimer's disease. And structurally, so once that gene, that APOE gene, that APOE4 gene, has been transcribed and translated, the protein that's produced structurally differs from the other two isoforms. There's an amino acid substitution, and that's due to a mutation in the APOE genetic sequence of our DNA. Okay, and that mutation results in a substitution of the amino acid that's being sequenced within the protein, and that substitution alters its overall conformation. So there's a slightly different shape. It has a slightly different overall quaternary structure, if you like, to the other two isoforms, and that impairs its interaction and its binding. And that essentially reduces its that essentially has many downstream effects. So one of them in particular is it plays a role in glial-mediated phagocytosis, as I said before, and that's associated with plaque clearance, because those microglia, they're going to try and clear those plaques, those extracellular plaques, and to do that they need the alipoprotein E, and the alipoprotein E4 isoform isn't as effective as doing that because of that amino acid substitution and that change in quaternary structure. Because these enzymes and proteins, they're incredibly specific, they're structurally specific, they're dependent on their quaternary structure to kind of fit their substrate or to fit what they're binding to, and that mutation makes it ineffective. So, and it makes it ineffective at synaptic maintenance and cell trafficking and glial-mediated phagocytosis, which is the main one, and it reduces plaque clearance. So you're going to get more of a plaque buildup and less of a plaque clearance. So Alzheimer's actually has a very strong inflammatory component. Like with most chronic inflammatory conditions, something we're realizing more and more as we progress through medical research is that a lot of diseases are not only perpetuated by inflammation, but they're also partially initiated by inflammation, and that alongside genetic risk factors, lifestyle, and all that stuff. But essentially neuroimmunology in particular, neuroinflammation, plays a massive role in kind of perpetuating this disease and making it more progressive and very challenging to treat. So as I said before, those soluble amyloid beta oligomers essentially govern downstream tau hyperphosphorylation indirectly through the initiation of pro-inflammatory signaling by our microglia and by our astrocytes. Now, those soluble amyloid beta oligomers, or those amyloid beta aggregates, we'll call them, they act as damage-associated molecular patterns, or DAMPs, and our microglia in particular, and our astrocytes to a lesser degree, are able to detect these DAMPs through toll-like receptors, CD3 and TREM2 receptors, and once those receptors, or once those DAMPs are detected, it initiates inflammasome signaling. So in the microglia, they activate this kind of cytosolic multi-protein complex referred to as the NLPR3 inflammasome complex, and once that's activated, you get an upregulation in gene expression or in the expression of pro-inflammatory cytokines, and they undergo a transcriptional shift themselves. They go from a phenotypic state into a pro-inflammatory state, if you like, and they're going to release pro-inflammatory cytokines like tumor necrosis factor alpha, interleukin 1 beta, interleukin 6, and interferon gamma. And those pro-inflammatory cytokines, like interleukin 1 beta, might stimulate the activation or overactivity of certain enzymes like JSK3B or P3A, mitogen activated protein kinase, and those enzymes are in particular associated with tau pathology, the hyperphosphorylation of tau, which is a generation of those neurofibrillary tangles. And in later phases of the disease, once those intracellular neurofibrillary tangles have built up, they will also act as DAMPs themselves. But what's essentially happening here is all of these processes, it's like an over amplification loop and like a positive feedback loop. So those plaques are going to initiate inflammation, but they're also going to perpetuate inflammation, and that inflammation is going to favor the amyloidogenic processing of the APP protein, as we said before, like tumor necrosis factor alpha and the activation of the pro-inflammatory cytokine tumor necrosis factor alpha. When it binds to that neuron, is going to activate those transcription factors that are going to suppress ADAM10 gene expression but upregulate or facilitate base 1 expression, so you've got more beta secretase secretase activity. Inflammation is going to continue to stimulate cytokine-mediated endocytosis of not just APP proteins, but various proteins expressed in the transmembrane of these neurons. It's going to create a more acidic environment, which is favouring enzymes like beta secretase, and it's favouring that pathological cleavage or amyloidogenesis, the formation of those amyloid beta 42 aggregates. You're going to get more oligomerization, you're going to get more plaque buildup, and you're going to get more pro-inflammatory signaling, more activation of kinase enzymes, more hyperphosphorylation, more buildup of neurofibrillary tangles, and it's just going to go round in circles, and the plaque's going to get bigger, the tangles are going to get more obvious, and eventually you get, unfortunately, you're going to get excitotoxicity, you're going to get increase in intracellular calcium in our neurons. It's going to activate dangerous enzymes like caspases, and you're going to get apoptosis and cell death eventually. And that's what you see when you look at later phases of the disease. You see significant brain atrophy, tissue atrophy. That's a reduction in certain areas, especially like localized atrophy in where the plaque buildup and the intracellular tangles are. And you see that in Alzheimer's disease, unfortunately, and that's one of the things that I think makes it so challenging to treat and understand, this kind of positive feedback loop, if you like, and that's with many kind of pro-inflammatory diseases. Like when you look at multiple sclerosis as well, a lot of diseases we get in our peripheral, like when you look at IBD, IBS, a lot of it, unfortunately, are these autoreactive immune cells just keep signaling, and they keep encouraging damage. Like in IBD, in IBS and all that, you can get stomach ulcers. In multiple sclerosis, you get that demyelination, and in Alzheimer's, you get this buildup of plaque and that buildup of intracellular neurofibrillary tangles, and they just keep kind of getting worse. Now, these pro-inflammatory cytokines, as we've basically just discussed, they act in both autocrine and paracrine fashions on both neurons and astrocytes. So those microglial cells activate astrocytes, they go into astrogliosis, they also release reactive oxygen species, pro-inflammatory cytokines. Our microglia will signal the neurons, they're acting on the neurons, and the neurons undergo transcriptional shifts. They're changing their on a transcriptional level, like as I said before, with ADAM10 and ADAM17, those genes become suppressed, and certain genes like BACE1 become upregulated. And as you know, BACE1 is favoring that amyloidogenesis because it's coding or translating that enzyme beta secretase, and beta secretase is going to cleave at the N-terminus of those APP proteins, especially if they're trafficked in those endosomes. That acidity favors its catalytic efficiency. It's more efficient as a catalyst at the pH. I think it's a pH of about 5. It's not an overly acidic environment, but that slight change in pH favors beta secretase activity. And then obviously beta secretase favors amyloidogenesis because you get that generation of the amyloid beta 42. They oligomerize and the plaques just keep building up, and the signals keep essentially perpetuating. The first pro-inflammatory cytokine we'll talk about, and this is the pro-inflammatory cytokine, just to kind of reiterate, that's been secreted by our microglia cells and also our astrocytes when they undergo astrogliosis and they undergo that transcriptional shift. Okay, these microglia and astrocytes, they're upregulating the expression of these pro-inflammatory cytokines, and tumor necrosis factor alpha is one of those cytokines, and it's a candidate for so many inflammatory conditions. It's used as a biomarker along with interleukin 6, especially to kind of diagnose and to kind of look out for inflammation. It's used as a biomarker for inflammation and chronic inflammatory diseases. So tumor necrosis factor, once it's secreted by our microglia, okay, so they've detected that our damage associated molecular patterns, which are our plaque, okay, and we got our plaque through our abnormal beta secretase activity, okay, and that resulted in the formation of these amyloid beta oligomers or plaques. Okay, so once our microglia detected that, it will activate its inflammasome signaling and it will upregulate tumor necrosis factor alpha. Once tumor necrosis factor alpha is secreted, it will bind to receptors on neurons. So neurons express in low densities, typically in physiological conditions, what's called TNFR1 receptors, and TNFR1 receptors, unlike TNFR2 receptors on neurons, are associated with less neuroprotective kind of mechanisms and more kind of pro-inflammatory mechanisms. And of course, more tumor necrosis factor binding will lead to an upregulation and in the expression of TNFR1. It will increase the density of TNFR1 expression in neurons as well. So that's another kind of transcriptional shift that you're going to see as tumor necrosis factor alpha is being secreted in high levels from microglia and our astrocytes. So anyway, tumor necrosis factor alpha will bind to TNFR1 on neurons, and this results in a cascade of intracellular reactions like most cell signaling. So it leads to the activation or the recruitment of adaptor proteins like TRAD, TRAF, and this eventually results in the activation of the IKK complex. And what you get from this is the activation of our nuclear factor kappa light chain, or NFKB, which is a transcription factor. Now, transcription factors directly can influence gene expression. They translocate to the nucleus where our DNA is, and they will alter they will alter the expression of some. They'll facilitate the expression of some genes and maybe suppress the expression of others. And in this case, our NFKB transcription factor, when it translocates to the nucleus, it upregulates base 1 gene expression. And as you know, the base 1 gene, once transcribed and translated, gets the beta secretase enzyme, so it codes for beta secretase. So now we have an upregulation in beta secretase gene expression, so more beta secretase, and of course this kind of inflammatory signaling is also leading to more, cytokine mediated endocytosis or endosomal trafficking of our APP protein. So we've got bases expressions upregulated, but it's catalytic efficiency is favored at that pH of the increased endosomal trafficking as well. So everything that's happening is favoring beta secretase, and alpha secretase is now being outcompeted, if you like, because its gene expression isn't upregulated. It's almost suppressed, and its activity is not optimum at that pH, unlike beta secretase. So everything that's happening is favoring amyloidogenesis and further buildup of extracellular plaques. Now, tumor necrosis factor alpha, as I said before, also increases AMPA receptor internalization. Okay, and AMPA receptors are also really important in neuronal signaling. Okay, and it increases endocytosis of many transmembrane proteins that are all involved in signaling for neural outgrowth, synaptic plasticity, just basically all the processes that speak for our central nervous system. So the next pro-inflammatory cytokine we'll talk about is interleukin-1 beta. Now there's a lot of information on these slides because I feel like it's necessary, so feel free to pause it and kind of copy it down. But interleukin beta is another one of those pro-inflammatory cytokines that have been secreted by our microglia if we're kind of telling this kind of story. And of course they can be secreted by astrocytes as well, but we're using the microglia are renowned for their kind of like toll-like receptors and detecting these damps. So when microglia respond to our damps or our plaque, what's going to happen, as I said before, is you get the activation of the cytosolic NLPR3 inflammasome complex. Okay, and that's a two-step multi-protein complex, and that will essentially upregulate the expression, as I said before, of many pro-inflammatory cytokines, including interleukin-1 beta. Now, both astrocytes and neurons possess the IL-1RI receptor, or the interleukin-1b receptor. Now, and its respective ligand, of course, is interleukin-1b. Now, the consequences are slightly different. Ligand binding in neurons and in astrocytes, the cellular consequences are slightly different. So in neurons, when it binds to the interleukin-1b receptor, so once interleukin-1 beta binds to those interleukin-1b receptors on neurons, it results in the activation of the MyDA8 dependent signaling cascade. Okay, and that's, a lot of you know, what is associated with the activation of our P3A mitogen activated protein kinase, and that's an enzyme that's associated, that serves many important functions, especially within the nervous system. In the central nervous system, it's involved in synaptic pruning, facilitating long-term depression long-term potentiations, the process in which we form new memories, and that involves the glutamatergic system along with their respective NMDA receptors. But in order to kind of refine those circuits, to form and refine those neural circuits, you also need not just long-term potentiation, but you need a balance of long-term depression as well to kind of prune those old circuits, to kind of refine them. And that's where P38 MAP kinase comes in. It's really important in that. But in the pathology of Alzheimer's disease, its overactivation obviously isn't good, because you get this kind of imbalance of long-term depression versus long-term potentiation. So you're going to get more synaptic pruning and less long-term potentiation, less synaptogenesis, less formation of new synapses. And also P38 MAP kinase is one of the primary candidates for tau hyperphosphorylation. And tau hyperphosphorylation, as you know, when tau becomes phosphorylated, it mislocalizes into the somatodendritic space, and it aggregates into those neurofibrillary tangles. So P38 MAP kinase is a prime enzyme and a really important molecular link between amyloid and tau pathology. And interleukin 1 beta is that kind of like indirect initiator of P38 MAP kinase. And interleukin 1 beta was secreted and its expression was upregulated because of the presence of the amyloid beta plaque. So you can kind of see how plaque pathology is governing downstream tau hyperphosphorylation. So P38 MAP kinase is a very, important enzyme in Alzheimer's pathophysiology. Now, in astrocytes, when interleukin 1b binds to those receptors, the IL-1B receptors in astrocytes, it also activates P3 MAP kinase, and that's correlated with a shift, if you like, a shift from their home. Astrocytes have their kind of homeostatic phenotype, and they shift into what's called reactive astrogliosis, and that's referred to as their damaged control state. And they will also release for certain inflammatory cytokines like interleukin 6, tumor necrosis factor alpha, nitrous oxide, the reactive oxygen species, and prostaglandins, which will amplify and exacerbate inflammation, because the astrocytes will also those pro-inflammatory cytokines and reactive oxygen species will also damage the neuron. Astrocytes also get compromised in this process, which I'll talk about later. But P38 mitogen activated protein kinase, it's that stress activated serine threonine kinase, and that isoform in neurons is associated with cellular danger signals as well, and synaptic pruning. And when it's overactivated, it has many consequences. It's got many cellular consequences, one being a higher rate of long-term depression. So we've got less synapses, or we're encouraging the formation of less synapses, less neural outgrowth. You get you lower the threshold for cell apoptosis, and also you get your tau hyperphosphorylation. So our P38 mitogen activated protein kinase, or P38 MAP kinase, is a really crucial enzyme in kind of perpetuating Alzheimer's pathology. And I thought, I'll do a slide on it to kind of help you understand why. Now, at the synaptic level, under normal physiological conditions, P38 MAP kinase is responsible for suppressing long-term potentiation whilst enhancing long-term depression. And when I say long-term depression, I don't mean depression in the psychiatric sense. I mean long-term depression is the kind of pruning, of old synapses, and long-term potentiation is the formation of new synapses, or new nerve connections. And that kind of speaks to the plasticity of our central nervous system. In order to achieve that kind of refined reforming of neural circuitry as someone gets older, you need old synapses to be eliminated, or new ones to be formed, and that kind of that facilitates efficiency of kind of nerve impulse, or nerve communication, if you like. Long-term potentiation itself is the formation of new synapses, and that relies heavily on the glutamatergic system and NMDA receptor, and it's their respective NMDA receptors. And essentially, when glutamate binds to those NMDA receptors, it triggers an intracellular kind of influx of calcium into the presynaptic neuron. And if that is sufficient enough, if that calcium influx is sufficient enough, and you get hyperpolarization, you kind of get the reform. You get the forming of a new synapse. There's a lot more complicated than that, and I'll go over it when I talk about memory and learning. And long-term depression is essentially the antithesis of that process, and it's the kind of elimination and pruning of synapses that are no longer serve us benefit. And that speaks to the process of learning itself. You know, as you get older, as I said before, as you're developing, you need to achieve, you know, healthy brain plasticity, you need a balance of long-term potentiation and long-term depression. So long-term depression isn't necessarily a bad thing. It's crucial in refining neural circuitry. But in the case of Alzheimer's, when P38MAPK is overactivated, you're going to get enhanced long-term depression, and you're going to get suppressed long-term potentiation. So synapses and nerve connections are going to get eliminated at an overt rate. And this is attributed to the cognitive dysfunction in Alzheimer's disease, and it's that you get way before significant neuronal loss. So even before those nerves are being, you know, even before apoptosis is happening in tissue atrophy, you get a significant decline in cognitive function even in early phases of Alzheimer's. And a lot of it is because of P38MAP kinase. You're going to get a lot of long-term depression. So a lot of synapses are going to be, you know, pruned, if you like, and it's going to affect that circuitry. It's going to disrupt that circuitry, and you're going to get a decline in cognition. Now, it is essentially this kind of feed-forward amplification loop, as I said before. Yeah, like I said, that interleukin-1 beta that's secreted from microglia will bind to those IL interleukin-1 beta receptors on astrocytes, and it results in increased expression, upregulation of P38MAPK in our astrocytes. Now, when astrocytes have an increased amount of P38 MAPK in particular, they go into that astrogliosis. So while they're in that state, that not only are they releasing pro-inflammatory cytokines and reactive oxygen species, they're also their astrocytes typically they're responsible for mopping up glutamate. As I said before, glutamate is a really important neurotransmitter in long-term potentiation. Okay, the formation of new synapses. Now, when astrocytes compromised in their glutamate uptake, what happens is you get prolonged exposure of nerves or neurons to glutamate, the neurotransmitter glutamate, and this can lead to excitotoxicity. So P38MAPK is indirectly linked to excitotoxicity that you see in Alzheimer's disease. So at the astrocytes, they're going to be unable to kind of mop up that glutamate, if you like, and you're going to get prolonged exposure of glutamate to those nerve cells, and those NMDA receptors are going to kind of stay open, and you're going to get an inappropriate influx of intracellular calcium. And when you get that inappropriate influx of intracellular calcium, it leads to excitotoxicity, and that essentially lowers the threshold for cell apoptosis as well, because high levels of calcium into a cell, as most people will be aware, leads to the activation of certain enzymes like caspases, which are associated with apoptosis. And essentially P38MAPK is essentially indirectly lowering the threshold for programmed cell death, and it's sensitizing neurons to apoptosis, and you see neuronal loss in later phases of the disease. Okay, and that when you get apoptosis happening on a massive scale, you're losing a lot of neurons that can't be replaced, and that's detrimental to one's health, one's cognition, okay, one's ability to think. So MAP kinase, or P38 MAP kinase, is at the center of this kind of self-reinforcing pathological loop. Okay, so, and not only that, but it's also responsible for tau hyperphosphorylation along with JSK3B and a few other kinase enzymes. But P38 MAP kinase is responsible for tau hyperphosphorylation as well. And tau hyperphosphorylation results in dendritic mislocalization, you get aggregation, and you get the formation of those neurofibrillary tangles. So you can kind of see P38 MAP kinase is playing many roles here, and all of them are favouring this kind of pro-inflammatory hostile environment in the central nervous system. It's making the tau hyperphosphorylation more worse, and you're getting more aggregation of neurofibrillary tangles. It's preventing or leading to excitotoxicity, because it's when it's upregulated in astrocytes, they're going to be in astrogliosis. They're not going to be mopping up glutamate because they're in their pro-inflammatory state. So you've got prolonged exposure to glutamate of nerve cells. You've got an increase of intracellular calcium now, and that's going to activate your caspases and your calpains. Okay, you're lowering the threshold for cell death or apoptosis, and high levels of P38 MAP kinase are also favouring long-term depression whilst suppressing long-term potentiation. So the next pro-inflammatory cytokine we'll talk about, which is secreted by both astrocytes and microglia, is interferon gamma. Now, interferon gamma is associated with the when it's secreted, okay, either in autocrine or paracrine fashion, it's associated with the upregulation of what's called the major histocompatibility complex, okay, and in neurons it's class 1, and in astrocytes and microglia, or in microglia, it's class 2. Now, in a lot of you might be aware, major histocompatibility complex is involved in antigen presentation, okay, and this is really important in later phases of the disease, because in later phases of the disease you get peripheral T cell or monocyte infiltration and they also end up over amplifying this kind of pro-inflammatory response. But this kind of upregulation of major histocompatibility complex class 2 allows for these microglia to act as antigen presenting cells. So when in later phases of the disease, when our peripheral T cells have infiltrated into our central nervous system, okay, those microglia are going to be presenting tau associated peptides and amyloid beta fragments, if you like, to these infiltrated T cells, okay, and those infiltrated T cells then will differentiate into Th1 and Th17 cells, and they're potent inflammatory signalers, okay, and they're going to over amplify they're going to over amplify this inflammatory response. So not only we've got our neuron, we had our microglia, and then we had our astrocyte, and now we've got our infiltrated T cells from the periphery, those monocytes, those CD4 plus cells, okay, and when they differentiate into our Th1 and Th17 cells, you're going to get a lot of inflammation. So we've introduced a new subset of cells, and this is typically in the infiltration occurs in later phases of the disease as the blood-brain barrier becomes compromised because of pro-inflammatory cytokines like interleukin-6, and the interferon gamma itself is associated with the upregulation of major histocompatibility complexes in both neurons and microglia. And in neurons, under normal physiological conditions, major histocompatibility complex class 1, it's typically expressed at very low levels, but there's an upregulation in the expression of the major histocompatibility complex class 1 in neurons because of interferon gamma release via those microglia. So I'll just briefly talk about interleukin-6 as well. So interleukin-6 is another pro-inflammatory cytokine. It's a pleiotropic pro-inflammatory cytokine, as with most of our other pro-inflammatory cytokines we've spoken about. Now, it's sometimes associated with neuroprotective mechanisms, but in the case of Alzheimer's disease, unfortunately it isn't. It facilitates T cell infiltration because, as I mentioned before, that leaky blood-brain barrier, those endothelial cells, they'll receive kind of interleukin-6 signaling from those astrocytic end feet that sit on the other side, or the central nervous system side of the blood-brain barrier, okay, and that signaling results in the upregulation of certain adhesive proteins like VCAM1 and those integrin proteins, and those proteins in the blood-brain barrier are associated with T cell adhesion. When you get an upregulation of, say, VCAM1, you're going to get more T cell adhesion from the periphery, and that's going to encourage T cell infiltration or monocyte infiltration into the central nervous system, and that's something that you definitely don't want. And that's not just seen in Alzheimer's, that's also seen in neurological conditions like multiple sclerosis. You get that T cell infiltration, and usually the kind of pro-inflammatory signaling that's going on in the central nervous system is going to cause an upregulation, mainly through interleukin-6. It's going to cause an upregulation in the expression of those proteins like VCAM1 in those endothelial cells that line the blood-brain barrier. Interleukin-6 is also playing the kind of similar roles as well as tumor necrosis factor alpha and interleukin-1b, in the sense that, you know, in neurons it's activating that JAK or STAT3 pathway, so the activation of our nuclear factor or an NF-κB factor that's going to influence or cause transcriptional shifts in gene expression. It's going to favour, say, base1 and reduce ADAM10 gene expression. So as you know, base1, it will favour the upregulation of the expression of beta secretase while simultaneously down-regulating the expression of alpha secretase. Okay, so that kind of links back to our amyloidogenesis, okay, and our plaque buildup is going to keep happening, and it's also causing, it's also perpetuating, if you like, that kind of that positive feedback loop, that amplification loop. A lot of these pro-inflammatory cytokines, they're causing transcriptional changes in nerve cells, but IL-6 in particular is the kind of candidate for causing that upregulation in VCAM1 density in endothelial cells that line our blood-brain barrier. And interleukin 6 also, I've mentioned it in the PowerPoint as well, it reduces astrocyte glutamate clearance, and that's through these kind of changes we're seeing, these intracellular changes that say favour astrogliosis as opposed to its normal, phenotypic astrocytes. So I just want to touch on this excitotoxicity a little bit more, and this kind of dysregulation of the glutamatergic signaling, because it is really important in kind of understanding, downstream effects that pro-inflammatory cytokines, this neuroinflammatory signaling, has on the disease itself. So glial cells and astrocytes, right, they are immune cells of the central nervous system. But they don't just, they also play roles that kind of contribute towards synaptic plasticity and neuro outgrowth, mopping up neurotransmitters. They're also able to signal to signal neurons in normal physiological conditions. They play other functions. They're not just immune cells, and they're not just responsible for like phagocytosis, and they're not just responsible for releasing pro-inflammatory cytokines. They have a lot of functions, and one of them is in astrocytes in particular, in reducing or glutamate-induced excitotoxicity. So most neurotransmitters, when they're released or secreted via exocytosis from the presynaptic cleft, they're broken down or degraded by enzymes to prevent prolonged exposure or overexcitation, hyperpolarization, or too much inhibition, depending on whether it's an excitatory or inhibitory neurotransmitter. Glutamate is an excitatory neurotransmitter, which means it will most likely cause that kind of depolarization, that change in electrochemical gradient, which will initiate a nerve impulse. Okay, and once glutamate is kind of finished serving its function, there isn't an enzyme that breaks it down. So astrocytes are responsible for mopping up that glutamate to prevent that prolonged exposure. Now, glutamate binds to those NMDA receptors in postsynaptic neurons, and that's the N-methyl D-aspartate receptors. And when upon binding, those channels open, if you like, and they allow for that intracellular influx of calcium, which is really important. That the calcium influx is really important. They're not just associated with excitotoxicity. But when amyloid beta oligomers impair glutamate uptake, but P3 MAP kinase in astrocytes also impairs glutamate uptake. So everything that's happening is kind of is working against this glutamate uptake. And when astrocytes are in their astrogliosis phenotype, their ability to mop up glutamate is also compromised. Now, what essentially happens is you have prolonged exposure to glutamate, and you have a increase in intracellular calcium influx, because glutamate is going to keep binding to those NMDA receptors and allowing for too much calcium, and you'll get calcium toxicity. This triggers the activation of calcium dependent enzymes like calpains and caspases, which are associated with apoptosis. So we're lowering the threshold for apoptosis. Okay, and this influx of calcium also overwhelms the neuronal buffering capacity. It triggers mitochondrial dysfunction. It prevents or interferes with oxidative phosphorylation, which is a stage of aerobic respiration, and that in itself, that disruption of oxidative phosphorylation leads to the generation of radical oxygen species, or reactive oxygen species like hydroxyl radicals, superoxide, and that's not good either, and I'll talk about that in the next slide. But essentially, it's this compromise in astrocytes, or this change in astrocytes and change in the kind of environment of the central nervous system. One of the results is glutamate induced one of the downstream consequences is glutamate induced excitotoxicity, because astrocytes aren't able to mop that up. Okay, you get prolonged exposure to glutamate, and you get that heavy influx of calcium, and you get that kind of you get all those negative effects. You overwhelm neuronal buffering capacity, you trigger that mitochondrial dysfunction, and then you lower the threshold for cell apoptosis. So the sustained activation of those extrasynaptic NMDA receptors via prolonged glutamate exposure, because of those, compromised astrocytes, is going to lead to that inappropriate influx of intracellular calcium into the neuron. Okay, and that interferes with neuronal buffering and processes like mitochondrial respiration or aerobic respiration. So it directly interferes with ATP production, and it particularly interferes with the electron transport chain and oxidative phosphorylation, which is where the majority of ATP is actually produced in aerobic respiration. Now, this interference leads to the generation of reactive oxygen species like hydroxyl radicals, hydrogen peroxide, and superoxide, but it also leads to the generation of nitric oxide, so reactive nitrogen species as well. And I want to reiterate here as well, it's not just through excitotoxicity that we get the generation of reactive oxygen species. It's also through, glial cells like microglial and astrocytes will also release reactive oxygen species. And when there's sustained inflammatory signaling, many glial cells will upregulate their expression of certain enzymes like nitric oxide synthase. So they're going to be, synthesizing and secreting nitric oxide a lot. And nitric oxide will react to oxygen, or reactive oxygen, to produce what's called peroxynitrate, and that's a highly toxic nitrogen species that damages lipids, DNA, it damages various proteins, and it damages proteins that are actually essentially might essentially be important in alleviating Alzheimer's pathology. So again, we're kind of, everything that's happening is perpetuating our kind of and leaning towards favoring the kind of like Alzheimer's or the pathological pathway, the pathophysiological pathway. Now, like peroxynitrate and these reactive oxygen species and nitric oxide will actually oxidize enzymes like protein phosphatase 2, which is a really important enzyme, or PP2A is a really important enzyme in reducing tau hyperphosphorylation, okay, under normal physiological conditions, which I'll talk about in later slides when we talk about tau pathology. In normal physiological conditions, protein phosphatase 2A will remove those, dephosphorylate or remove those phosphates from tau, allowing it to reassociate to its microtubule and facilitate neural outgrowth, okay. And protein phosphatases 2A contain that serine, threonine amino acid sequencing in their active site, and that's susceptible to oxidative damage. And when that damage happens, their catalytic efficiency is reduced, okay, and they're getting outcompeted by kinase enzymes who are hyperphosphorylating the tau protein and encouraging that aggregation, that formation of neurofibrillary tangles. So just to reiterate, okay, the key takeaway is that excitotoxicity through glutamatergic signaling or sustained glutamatergic exposure and NMDA receptor binding leads to an influx of intracellular calcium. This interferes with neuronal buffering, and this intracellular calcium will also interfere with processes that are happening in the mitochondria, like oxidative phosphorylation, and that's really important in energy production. Okay, and then those processes are going to be interfered, and they're going to generate reactive oxygen species, which are going to oxidize and damage structures within the cell. And we've also got reactive oxygen species being secreted and reactive nitrogen species being secreted from our microglial cells and our astrocytes, okay, because this inflammatory signaling, this inflammatory environment is leading to the upregulation of enzymes like nitric oxide synthase in glial cells. So you're going to get more nitric oxide, and eventually you get the formation of peroxynitrate when nitric oxide reacts with reactive oxygen, okay, and that peroxynitrate is a potent oxidizing agent. It damages lipids, it damages DNA, and it damages like enzymes like protein phosphatase 2A. So I'll just very briefly touch on the role of the blood-brain barrier, or the BBB as it's sometimes referred to, in Alzheimer's disease, and we'll touch on this more in multiple sclerosis, I think. So the blood-brain barrier is essentially this kind of protective interface of endothelial cells and pericytes. On the central nervous system side, it's got those astrocytic end feet that can kind of communicate with those endothelial cells, and it's essentially its function is to maintain and promote central nervous system homeostasis. The central nervous system is a highly specialized, highly sensitive system, and the blood-brain barrier helps regulate what's going into the central nervous system. Okay, so it doesn't disrupt that homeostasis and cause massive damage, which is what we will see later on in Alzheimer's disease. Now, the blood-brain barrier, I like to think of it as acting almost like a phospholipid bilayer in a cell membrane, just on a more vascular level. Okay, and essentially, under normal physiological conditions, for example, monocytes like CD4 plus cells, our T cells, aren't typically found in the central nervous system. Okay, as I said before, the astrocytes and the glial cells are our predominant immune cells in the central nervous system, and that's because of the blood-brain barrier. The blood-brain barrier is stopping those kind of cells from getting into the periphery. It's stopping harmful substances that are probably harmful to the central nervous system, okay, but not as harmful to our peripheral system, from getting into the central nervous system. But in later phases of many neurodegenerative diseases like Alzheimer's and like MS, the blood-brain barrier becomes leaky. So you get like a leaky blood-brain barrier, and it allows for the infiltration of cells like T cells from the periphery into the central nervous system. Now, this is actually to do with increased inflammatory signaling. As I mentioned before, interleukin 6, for example, high levels of interleukin 6 being signaled from the central nervous system to those endothelial cells that kind of line the blood-brain barrier will result in, as I said, these signals change cells on a transcriptional level. So these endothelial cells are going to go through a transcriptional shift, and they're going to upregulate adhesive protein receptors like the VCAM1, and VCAM1 expression promotes adhesion from circulating monocytes and T cells. Okay, so it's promoting that cellular adhesion, and that allows for the infiltration. So VCAM1 and integrins in multiple sclerosis and in Alzheimer's allow for T cell infiltration into the central nervous system, and then that's when you get, once T cells infiltrate into the central nervous system, okay, they're going to differentiate into those Th1 and Th17 cells, and that's because of our, as I mentioned before, our interferon gamma secreted by our glial and astrocytes is going to encourage major histocompatibility complex expression and upregulation. Okay, so those T cells will respond to those major histocompatibility or MHC classes that are expressed on our microglia, expressed on our astrocytes, and they're going to differentiate into Th1 and Th17 cells, and they're going to contribute to this kind of inflammatory assault that we're seeing on our central nervous system. And typically, as I said before, in normal physiological conditions, VCAM1 is expressed in quite low densities in the endothelial cells. So T cell adhesion from circulating T cells, monocyte adhesion from circulating kind of monocytes in our periphery is reduced, and you don't get any infiltration. But in diseases like Alzheimer's, you see a lot of T cell infiltration, a lot of peripheral, T cells infiltrating from the periphery into the central nervous system. So now I'm going to talk a little bit more about tau pathology. So tau itself is a protein that's expressed in neurons, and it's essentially, its function is to stabilize and bind to those microtubules. Okay, and those microtubules are large components of the cytoskeleton you see in eukaryotic cells. Okay, and neurons are no different. And by binding to those microtubules, it can stabilize them and facilitate neuronal outgrowth, and that is critical in neuronal plasticity. Okay, because this is all these processes are contributing towards the reforming, refining, and pruning of neural circuits to get more efficient circuits. Okay, as we're learning, as our brain's developing, and under normal physiological conditions, tau binding or tau stabilizing or binding to those microtubules is regulated by kinase and phosphatase enzymes. So you need a balance of your phosphatase and kinase enzymes, okay? And those kinase enzymes will phosphorylate tau that's bound to the microtubule, and once tau becomes phosphorylated, it delocalizes or dissociates from the microtubule into the somatodendritic space. Okay, and phosphatase enzymes will come and then dephosphorylate that tau protein, allowing it to rebind, okay, and rebind to the microtubule and facilitate the movement and neuronal outgrowth. Now, as you know, in Alzheimer's pathology, we have an upregulation of kinase enzymes like P38 MAP kinase. We also have an upregulation or upactivity of JSK3B. Okay, so this means more phosphorylation is going to be happening than dephosphorylation. So there's a there's a higher amount of kinase enzymes than there is phosphatase enzymes. So you're going to get more phosphorylation. Okay, and when tau becomes phosphorylated, it's going to dissociate from that microtubule into the somatodendritic space, and it's more likely to aggregate, okay, and to form these kind of oligomers that will eventually become neurofibrillary tangles. So the phosphatase enzyme itself essentially can't keep up. It can't remove those, it can't dephosphorylate at the rate that the kinase enzymes are phosphorylating the tau protein itself. And of course, once you get this kind of neurofibrillary tangle formation, you get a destabilization of the cytoskeleton, because tau isn't able to bind when it's in that kind of phosphorylated state, and when it's in those tangles, in those kind of neurofibrillary tangles, it's unable to bind to the microtubule and facilitate neural outgrowth. So again, this kind of the plasticity, the overall central nervous system plasticity, okay, is compromised due to these mechanisms, due to these pathological mechanisms. Now, protein phosphatase A, the enzyme, which I'll talk about in the next slide, the enzyme that's involved in kind of dephosphorylating or removing those phosphates from the tau, allowing it to kind of rebind, is also kind of downregulated. Its expression is more downregulated in these inflammatory conditions, and it's also susceptible to damage to those reactive oxygen species. So it's being outcompeted by kinase, but it's also getting damaged. Those serine, threonine components, amino acid sequencing in its active site that it kind of attributes its catalytic efficiency to, become oxidized and they become damaged. And phosphatase 2A, it gets compromised, so its catalytic efficiency is decreased, and its expressions become downregulated. It's getting outcompeted by kinases. This is why you get what's called hyperphosphorylation. And essentially this then leads to microtubule instability, okay, and neural outgrowth is compromised and plasticity is drastically compromised. And this doesn't just affect neural plasticity and outgrowth. This also affects, organelle trafficking, vesicular migration. It has many downstream effects on the cell itself, many detrimental effects. So under normal, just to kind of recap, the main takeaway is that kind of imbalance between kinase enzymes and phosphatase enzymes is what we kind of attribute this tau hyperphosphorylation to. We've got our p38 MAP kinase that's been activated by tumor necrosis factor alpha, interleukin 1 beta. It's kind of upregulated its expression and activity of p38 MAP kinase, okay, and tau phosphorylation is going to be more tau phosphorylation is going to be happening, okay, and phosphatase is unable to kind of keep up. It's getting outcompeted, and you get that eventually you get aggregation and the formation of those neurofibrillary tangles, which are composed of those hyperphosphorylated tau monomers, if you like. As I said before, the protein phosphatase 2A, or often PP2A as it's seen in literature, is responsible for kind of removing those phosphate groups from tau and allowing for it to kind of rebind or restabilize with its microtubule, and that then facilitates neural outgrowth, okay, and neuronal outgrowth, which is really important in central nervous system plasticity. Now, it's a heterotrimeric holoenzyme, and it has that kind of dominant serine threonine sequencing in its active site that it attributes its catalytic efficiency to, and the subsequent excitotoxicity because of this kind of the compromise in astrocytes' ability to kind of mop up glutamate. Sustained NMDA signaling results in that kind of intracellular influx of calcium, okay, and that you get that excitotoxicity, you get the generation of these reactive oxygen species, and that oxidizes that serine threonine component on the phosphatase enzyme. It reduces its catalytic efficiency, it reduces its ability to kind of remove those phosphate groups. Its expression is downregulated as well because of our NF-κB transcription factors and because of our pro-inflammatory signaling. So it's getting outcompete, and everything's favoring kinase activity while not favoring phosphatase activity. Hence, we call it hyperphosphorylation. So because kinase is phosphorylating that tau, it's dissociating from that microtubule into the somatodendritic space, and then it's aggregating with other phosphorylated tau monomers to form these kind of neurofibrillary tangles, and it's a massive problem. And you can kind of see how upstream kind of like or downstream amyloid beta formation or amyloid or senile plaque formation in the extracellular space has kind of resulted in tau hyperphosphorylation downstream, because the reason our kinase enzymes are upregulated in expression and they're activated more is because of the pro-inflammatory signaling that's being the pro-inflammatory signaling from our glial cells, and they're responding to that buildup of amyloid plaque initially. The thing that kind of made them shift from their phenotopic state into their pro-inflammatory state was the detection of that kind of amyloid beta plaque. But yeah, the main takeaway, I guess, from these slides is that phosphatase is compromised because of the reactive oxygen species generated not only from the glial cells but also from the excitotoxic effects of, you know, calcium influx and glutamatergic exposure. They become oxidized and their catalytic efficiency is reduced. They're unable to kind of remove those phosphate groups, and that's really important. That kind of kinase phosphatase balance in a cell is really important in, you know, maintaining and regulating neurite outgrowth, and that whole process is completely skewed and compromised and favors the kinase enzymes more than it does the phosphatase enzymes, and that the consequence of that is that kind of tau hyperphosphorylation, subsequent mislocalization into the somatodendritic space, and then subsequent aggregation and oligomerization of those phosphorylated tau monomers. So now we've spoken about the kind of pathophysiology behind Alzheimer's, and I will do a recap at the end. We're going to briefly talk about pharmacological interventions in Alzheimer's disease. As I said before, there's no known cure for this disease. It is a progressive neurological disorder and no cure exists. But what there is drugs that will either suppress symptoms or work towards a reduction in pathology, which symptom suppressing medications will impact the quality of life, but they won't necessarily slow down disease progression and extend life. But these disease modifying therapies we'll talk about later, like the monoclonal antibody therapies, actually aim to clear the amyloid plaques in particular. And as you know, those amyloid plaques are really important in acting as that kind of inflammatory kind of initiator for our glial cells in our downstream tau hyperphosphorylation. So reducing those plaques essentially can reduce inflammation, and a reduction in inflammation and a reduction of MAP kinase activation might allow phosphatase to have a chance in kind of preventing the formation of those neurofibrillary tangles. But we'll kind of get into it and we'll have a look at treatments. So the first treatment we'll talk about is donepezil. And as I said before, this is more of a symptom suppressive treatment. So we're not targeting mechanisms that contribute towards amyloidogenesis or tau hyperphosphorylation. So we're not reducing plaque load, and we're not preventing the formation of those kind of intracellular tangles. But what we're doing is we're essentially suppressing some of the symptoms associated that are caused from these mechanisms in order to give the patient a better quality of life, really. So this won't extend the patient's life necessarily. It won't reduce disease load. But what donepezil will do is compensate for that loss in cholinergic signaling that is experienced during Alzheimer's disease, especially in later phases of Alzheimer's disease, when a lot of those cholinergic neurons are lost. They undergo, you know, apoptosis. They're lost and not replaced in the basal forebrain. And to compensate for that lack of cholinergic signaling, donepezil is administered. Now, pharmacologically speaking, donepezil is an inhibitor of the enzyme acetylcholine esterase. As many people will know, as many of you will know, acetylcholine esterase is an enzyme that breaks acetylcholine down into acetic acid and choline once it's been secreted into the synaptic cleft via the presynaptic neuron, and it's exerted its effect, its chemical effect on the postsynaptic neuron, okay, in order to prevent prolonged exposure. Now, I would assume most of you are familiar with the concept of synaptic transmission, especially if you're a kind of first or second year life science or medicine student. But for those of you who aren't, I'll quickly just kind of speed roll through it, just so everybody's on the same page. So you're in signaling, okay, in the terms of the central nervous system, okay, and also our peripheral system, but we'll talk about the central nervous system. Synaptic neurotransmitters allow for neuronal communication. They allow for one neuron to send a signal to another, and depending on the type of neurotransmitter, depending on the receptor that it binds to, will depend on whether or not that neuron will carry on a nerve impulse or inhibit a nerve impulse. In the case of acetylcholine has both excitatory and inhibitory effects on the central nervous system, depending on what subset of receptors it binds to. Now, let's say the presynaptic neuron releases acetylcholine, all right, via those little vesicles of the vesicular migration to the membrane of the presynaptic neuron, and then the acetylcholine is released into the synaptic cleft via exocytosis, and it will then bind to, say, nicotinic receptors. In the case of acetylcholine, nicotinic receptors that are expressed in moderate densities on the postsynaptic neuron, and then that will stimulate the flow of ions into the postsynaptic neuron. And depending on that electrochemical gradient and how electrochemical gradient, the charge of the ion, and the amount of depolarization that happens, if you like, will depend on whether a nerve impulse is continued or inhibited. And donepezil essentially works by prolonging exposure of postsynaptic neurons to acetylcholine to compensate for that loss in cholinergic neurons that's seen in later phases of Alzheimer's disease. So what donepezil is doing is it's binding to acetylcholine esterase, and it's preventing the breakdown of acetylcholine into acetic acid and choline. Okay, so it's leaving acetylcholine in the synaptic cleft for longer. This results in prolonged exposure, which is what we want in the context of Alzheimer's disease, because a lot of those cholinergic neurons have been lost. Okay, and that's going to have effect on cognition, because not just glutamate, but acetylcholine also plays a really important role in working memory. Nicotinic receptors, they play a really important role in working memory. They also play, alongside glutamate. So that loss in cholinergic neurons has some kind of detrimental effects, especially on cognition. So by administering donepezil, you are essentially making up for that loss in cholinergic neurons. You're giving what little neurons remain a chance to kind of continue that kind of those cholinergic signaling circuits, if you like, and that's essentially through inhibition of the acetylcholine esterase enzyme. Now, in terms of evaluating these treatments, as I said before, this isn't disease modifying, so we're not targeting plaque load, we're not targeting any mechanisms that contribute towards amyloidogenesis, plaque formation, we're not targeting mechanisms that contribute towards tau hyperphosphorylation. All you're doing is inhibiting the acetylcholine esterase enzyme and leaving the acetylcholine intact in that synaptic cleft, allowing for that postsynaptic neuron to get sufficient exposure or prolonged exposure to acetylcholine to compensate for that loss in cholinergic neurons, especially in the basal forebrain, and that's seen in Alzheimer's patients in later phases of the disease. So the next therapy we're going to talk about is memantine, and this is a non-competitive NMDA receptor antagonist. Now, it's often given in combination with donepezil or other acetylcholine esterase inhibitors. And again, I'm talking about this as more of a symptom suppressive therapy. It's not a disease modifying therapy, because as we know, the core molecular hallmarks of Alzheimer's disease is that buildup of extracellular senile plaque, and then that kind of buildup of intracellular neurofibrillary tangles. And these therapies aren't targeting mechanisms that interfere with the processes like amyloidogenesis or tau hyperphosphorylation. So it's not considered a disease modifying therapy. But anyway, to kind of talk about memantine for a little bit, so we mentioned before the consequences of excitotoxicity. So and how an inflammatory environment and how a kind of Alzheimer's driven environment in the central nervous system can lead to impaired glutamate uptake. So we've got our active astrocytes, which under normal physiologic conditions will mop up that glutamate that's in the extracellular space in the central nervous system. But when they're in their kind of astrogliosis phenotype, when they're in their reactive phenotype, they're not going to be mopping up that glutamate as much. And to make things worse, the presence of that extracellular senile plaque, that amyloid plaque, is going to compromise what little glutamate uptake there is already. So even if there is some glutamate uptake taking place, it's being compromised by that senile plaque. So you've got prolonged glutamate exposure, and that leads to sustained binding, extra synaptic binding on NMDA receptors, which leads to a uncontrolled amount of calcium going into the cell, and that interferes with processes like oxidative phosphorylation, and you get the generation of your reactive oxygen species. Okay, and those reactive oxygen species will damage and oxidize enzymes like protein phosphatase 2A, which, as you know, is really important in counterbalancing the effects of our P38 MAP kinase and JSK3B and reducing tau hyperphosphorylation, okay, reducing those neurofibrillary tangles. So this excitotoxicity has extreme implications on nerve cell, on the nerve cell. It's got extreme cellular implications. Okay, and not only that, but this heavy kind of calcium exposure will activate certain enzymes like caspases, okay, and that will lower the threshold for apoptosis. But this is where memantine comes in. So memantine will non-competitively bind to those NMDA receptors, and it acts in an antagonistic fashion. Okay, so it's going to keep those receptors closed. It's going to prevent that influx of intracellular calcium, which is what we want. What it's not doing is promoting glutamate clearance and reducing glutamate exposure. All it's doing is binding to those receptors and keeping them shut, almost like a door, almost like a lock and a key, keeping it shut and preventing calcium from coming into the cell. Okay, so that reduces excitotoxicity, okay, and has some positive downstream effects. But it dampens excitotoxicity, but it doesn't, it in terms of like downstream, if you want to talk about its efficiency, it doesn't, it's not reducing disease load or plaque load. And yes, we get the generation of reactive oxygen species from that influx of intracellular calcium, but remember, those reactive oxygen species and reactive nitrogen species, or nitrosative species, okay, they're also being produced by our glial cells and our astrocytes. So you're still getting exposure to reactive oxygen and nitrogen species, even with the addition of memantine. And just to reiterate, when a patient is given memantine in combination with donezepil, what's happening on a kind of pharmacological level is you've got acetylcholine. You're trying to prolong or encourage cholinergic activity, and memantine is kind of simultaneously reducing that glutamatergic signaling via NMDA antagonism. So you're kind of reducing that excitotoxicity while also encouraging or kind of facilitating cholinergic signaling. And that's the kind of strategy that's used with that combination therapy, if you like. So now we're going to move on to disease-modifying therapies, and monoclonal antibodies are at the forefront of disease-modifying therapies, and you'll see them across pharmacology. They're more novel treatments, they're intravenous treatments, and they are seen that there's monoclonal antibodies now for all sorts of diseases like multiple sclerosis. You can see it for gastroenterological conditions. Monoclonal antibodies are being investigated and used as therapies for many inflammatory diseases, and aducanumab is one of those antibodies that is given or administered to patients with Alzheimer's, and it's considered an anti-amyloid monoclonal antibody. So it selectively binds to those aggregated amyloid beta oligomers and those mature plaques, and it encourages or facilitates microglial mediated phagocytosis. So it's actually encouraging plaque clearance from our glial cells. And you see this in patients when they have PET scans. You can actually see it's quite effective because it reduces plaque burden. And as we know, plaque burden or amyloid beta accumulation has downstream effects and essentially governs tau hyperphosphorylation indirectly through pro-inflammatory signaling from microglia responding to the presence of those plaques, and that in turn is they're secreting pro-inflammatory cytokines which are activating our kinase enzymes and our JSK3B enzymes and encouraging tau hyperphosphorylation and subsequent formation of neurofibrillary tangles. So that's why a lot of these monoclonal antibodies that we'll talk about, they kind of target this plaque pathology, because by targeting plaque pathology, you're reducing downstream tau hyperphosphorylation and promoting plaque clearance at the same time. Now, in terms of cognitive benefits, they're modest to little, so they're not amazing. And of course, and I think a lot of this is to do with neuronal loss. Once you've, especially if you've been suffering with Alzheimer's, all the mechanisms have been happening and for a while, okay, and you get you get that significant neuronal loss. This isn't working to kind of replace lost neurons. It's not like it's not a stem cell therapy. It's not anything like that. It's just reducing plaque clearance, and it does have some effects on cognition, but it's not, once those cells are damaged or lost and that you get that tissue atrophy, it's very hard to come back from that. So even aducanumab, being a great novel treatment that it is still not as effective in, it's effective in extending life and slowing down disease progression. But again, it's not stopping that kind of autoreactive immune response. It's not stopping core mechanisms that drive this disease. So lecanemab is another example of an anti-amyloid monoclonal antibody, and it works similar to the aducanumab, but it's more selective. It only binds to the protofibrils. So I mentioned before, when you get that kind of pathologic processing of APP, you get the formation of those amyloid beta 42 species, and they'll oligomerize into these kind of small protofibrils. Okay, and then with that oligomerization will keep happening until you get a mature plaque. Well, lecanemab binds to those protofibrils, preventing the formation of future mature plaques. But what it's not doing is binding to those big mature plaques. They don't target mature plaques, they target those protofibrils, and they prevent new plaque formation. Because of course, it's all well and good, promoting plaque clearance, but we still have a mechanism, a loop going on, into a pathophysiological loop, a pro-inflammatory loop going on, that's triggering increased endosomal trafficking of APP. Beta secretase expression is still upregulated, so there's still going to be new plaques forming, okay, at all phases of the disease. So this is essentially preventing those new plaques from being formed. And it works the same way. So when it's binding to those protofibrils, it's facilitating microglial mediated phagocytosis. So those microglia will kind of phagocytose those plaques, and they'll break them down, and it promotes that plaque clearance. Those protofibrils, it will target them and it will break them down, promoting clearance and promoting formation of new mature plaques. So the next therapy or treatment we're going to talk about is a disease modifying therapy, and it's a more novel therapy, and it's just come out of China. And it's a very interesting in the way it works, because mechanistically speaking, we didn't really speak about the role the gut microbiome plays in Alzheimer's. This is a more kind of novel kind of take on disease mechanism. Of course, the core pathology is still relevant, very relevant. The whole neuroinflammation, the plaque buildup, the tangle, downstream tangle buildup. But sodium oligomanate kind of takes a kind of different approach. It targets the gut-brain immune axis, okay, and this essentially reduces peripheral inflammation and subsequent T cell infiltration into the central nervous system. Essentially, there's a lot of research on our gut microbiome that's being done, and dysbiosis or a dysregulation of our gut microbiome can lead to the secretion of inflammatory kind of peptides by our gut microbiome, because these bacteria that live in your gut, they're capable of producing peptides that can communicate with your enteric neurons. They can communicate with your peripheral system, and they have a bigger impact than anyone had ever anticipated on our biology itself. And essentially, how sodium it's orally administered, and how it's working is it's reversing Alzheimer's associated dysbiosis, and there's an imbalance in the microbiome that is associated with Alzheimer's disease that can encourage peripheral T cell inflammation and subsequent infiltration into the central nervous system. Now, sodium oligomanate remodels the gut microbiome, so it kind of gets rid of that dysbiosis, that disbalance, and it reduces the release of certain pro-inflammatory amino acids such as phenylalanine and isoleucine that are thought to contribute to T cell or Th1 cell differentiation, inflammation, and infiltration into the central nervous system. So by normalizing the kind of gut microbiome composition and reducing these metabolites, right, it suppresses Th1 cell expansion and limits their trafficking into the central nervous system, and it dampens that neuroinflammatory response. And this reduction in peripheral immune pressure leads to a decrease in microglial activation in the central nervous system, or overactivation. Okay, and it attenuates the release of inflammatory cytokines like interleukin 1 beta, interferon gamma, and you've got a reduced activation of downstream major histocompatibility complex expression, p38 MAP kinase expression, and as a consequence, it's essentially kind of dampening down that inflammatory loop. And it's quite clever how it's doing that, because it's doing it through the microbiome. So it just goes to show that our microbiome plays a much bigger role in driving and facilitating these diseases than people initially thought. And it kind of leads back to what I was saying earlier about diet. A lot of diets in the 21st century lead to dysbiosis, a dysregulation in your gut microbiome that can have massive implications on your health. And your gut microbiome has a lot of kind of hold more research is coming out to show it has a big hold over your system, your central nervous system. Your gut microbiome can communicate with your peripheral and central nervous system very efficiently. And of course, it's a kind of like, if you think about it, it's like a symbiotic relationship, really. Our gut microbiome is essentially a part of us. And, when that becomes disrupted, it can have severe consequences, and it's often linked to inflammation, but it's essentially kind of reframing systemic inflammation. Okay, and it's kind of at the forefront of novel kind of novel treatments, if you like. And it's not targeting amyloid plaques directly. It's essentially reframing um Alzheimer's pathology as this kind of systematic inflammatory disorder with like a gut kind of a dysbiotic gut immune kind of origin. Okay, and it's really interesting how this treatment's working, but it is still considered a disease modifying therapy, because as a result downstream of as a result of a reduction in neuroinflammation, you're gonna see a reduction in plaque buildup, because plaque buildup becomes dependent on the kind of neuroinflammation in later phases of the disease, because that neuroinflammation is kind of upregulating base1 expression, it's increasing APP trafficking, and of course that neuroinflammation is driving P38MAPK activation. So you will see a reduction in both amyloid plaque load and tau hyperphosphorylation. Again, it's unfortunately there's still no cure for Alzheimer's disease. I just want to make that very clear. But the therapies that are coming out are becoming more and more effective in disease modifying, and we're understanding more about inflammation. We're looking at it as a more of a systemic approach rather than a localized phenomena. So, like, it's not just, as I said, the inflammation is happening on a systemic level, usually in inflammatory diseases. Okay, but the area that's damaged, it is usually where it's most localized. Like, for example, if you're suffering with a neurological or neurodegenerative inflammatory disorder, you're going to think that the inflammation is happening in the central nervous system, but it's actually happening on a systemic level. Okay, because same with like gut inflammatory disorders like IBD, it's not just happening in the gut; it's happening on a systemic level. It's just kind of it's more localized to the gut, but it's still happening on a systemic level. So that kind of way of thinking might actually be very effective. That kind of thinking kind of outside the box and thinking of it from more of a systemic issue rather than a localized phenomena can actually is actually showing to be more effective in developing these kind of treatments. So it's really exciting. So we've spoken about therapies that kind of relate to amyloid or amyloidogenesis and plaque formation. But obviously tau hyperphosphorylation, yes, amyloid beta oligomerization and plaque formation, the formation of senile plaques does drive downstream tau hyperphosphorylation. But once tau hyperphosphorylation starts to happen, it also starts to act as a damp, and it also starts to become a problem. And it's something that they're looking at targeting. There is no, tau aggregation inhibitors, which I'm about to talk about now, are kind of more novel therapies that have not been approved for the treatment of Alzheimer's yet. But I think it's worth mentioning, because we've spoken a lot about the concept of targeting amyloid plaque, but there's also, therapies and strategies put in that are being developed right now to target tau aggregation, tau hyperphosphorylation, and also therapies that are being developed to compensate for tau loss, okay, and microtubule instability. And one of those is tau aggregation inhibitors. Now they essentially stabilize monomeric tau. So once tau's become hyperphosphorylated by the kinase enzymes, it will mislocalize into the somatodendritic space, and then it will aggregate into these kind of tangles. What LMTX is doing, and this is one of the examples of the tau aggregation inhibitors, LMTX is binding to those monomers, those phosphorylated tau monomers, and stabilizing them, preventing them from having the ability to aggregate and to oligomerize and aggregate into these big kind of intracellular tangles that you see. It's not preventing tau hyperphosphorylation though, so it's not inhibiting those kinase enzymes or those JSK3B enzymes that will phosphorylate the tau protein. Just once they've been phosphorylated, they'll delocalize from the microtubule, and then they'll try and aggregate, but this LMTX is just preventing that aggregation from happening. So the next set of therapies we're going to talk about is the anti-tau immunotherapies, and these are similar to the aducanumab in the terms of like they're antibodies that are targeting the hyperphosphorylated tau or the misfolded tau, and the obviously the aducanumab is targeting those antibodies are anti-amyloid beta antibodies that are targeting that and facilitating macroglial mediated phagocytosis of that amyloid plaque, which is the same principle for the anti-tau immunotherapy. Okay, those antibodies will target misfolded tau and facilitate microglial mediated phagocytosis, and you'll get, tau clearance. But the anti-tau therapy is much further along than the tau aggregation inhibitors, but they've still not reached the market yet. So anti-tau immunotherapy is still under investigation for safety, for efficacy, and even the diagram that's being shown or demonstrated there is from a transgenic mouse model, transgenic Alzheimer's mouse model, and looking at the effectivity of tau immunotherapy. But it's the kind of same principle as aducanumab, just targeting the hyperphosphorylated or misfolded tau as opposed to the senile plaque, and eventually you will see treatments like this as time goes on and they kind of move through those clinical trial phases. You'll see them reach hospitals and reach patients with Alzheimer's, targeting that tau pathology. So the next set of treatments that are currently being investigated, and I'd say they're way more in the works. They're way further behind than our tau aggregation inhibitors, and of course they're much further behind than our anti-tau kind of immunotherapies. But tau kinase inhibitors, or novel small molecules that aim to inhibit kinase enzymes like JSK3B, CDK5, P3-AMAP kinase, and bind to their serine threonine components, okay, to prevent the phosphorylation of tau. As you know, those neurofibrillary tangles, their formation is attributed to tau hyperphosphorylation, and that in itself is a result of an imbalance between our phosphatase enzymes and overactivation of our kinase enzymes. So we have too many kinase and a lack of phosphatase enzymes that's resulting in this kind of hyperphosphorylation and subsequent formation of these intracellular tangles. Now, it makes sense, right, to a small molecule, if you could develop a small molecule to bind to those kinase enzymes, that you'll prevent tau hyperphosphorylation and you'll prevent neurofibrillary tangle formation. While that's true theoretically, pharmacologically speaking, it's definitely not that simple, and that's why this is so still in the works, because obviously the main challenge is selectivity and off-target interactions. Okay, so kinase enzymes, we villainize them a lot in this kind of pathophysiology video, but kinase enzymes themselves are so important in essential, the fundamentals of cellular signaling and cellular pathways, for the most part, depends on these activity of kinase enzymes. So when they become inhibited, off-target inhibition has serious cellular consequences. You get toxicity, like drug toxicity, quite quickly with these kind of treatments, especially in animal models, as it's been shown. So off-target interactions is a massive issue with these kind of with inhibitors like that work like this. So this is why kinase inhibitors, the development of kinase inhibitors is still quite kind of, in Alzheimer's disease, is very far behind, if you like, because there's pharmacologists are trying to, how can we achieve treatment efficacy without getting those off-target interactions and subsequent toxicity? And that's one of the challenges currently with treatments like this being developed. So that's why they're a lot further behind in our kind of anti-tau immunotherapies, as well as our tau aggregation inhibitors. So preventing hyperphosphorylation of tau itself can be quite challenging. So a lot of these treatments that target tau pathology tend to either compensate, or a lot of these treatments that are targeting tau pathology that are kind of further along in development tend to either compensate for a loss of tau and stabilize kind of microtubules and facilitate neural outgrowth, or they'll prevent the aggregation of tau once it's been hyperphosphorylated, but they're not actually targeting the main driver, the main problem, which is that the phosphorylation of tau and the overactivation of kinase enzymes. But of course, if you're targeting kinase enzymes, you get, selectivity is a massive problem. So many kinase enzymes are really important in these signaling processes that inhibition will cause more harm than good and will lead to drug toxicity. But yeah, still in tau kinase inhibitors are still being investigated, and maybe eventually we'll be able to achieve that in pharmacology. But right now, that appears to be one of the main challenges. So again, microtubule stabilizing agents are kind of they're a lot further along than tau kinase inhibitors, but they're still a little bit behind on tau immunotherapies. But it's still kind of targeting that tau pathology, and it's not preventing the hyperphosphorylation, as we know that's a lot more complicated to do, because of those kind of pharmacological nuances with selectivity and off targets. But these microtubule stabilizing agents aim to compensate for tau loss. So like epithylone D and TPI-287 can directly bind to these microtubules, enhance their polymerization, and allow for that kind of neural outgrowth. So they're kind of compensating for the loss of microstability in the cytoskeleton that's induced by tau detachment. Okay, so this preserves axonal transport, the transport of all vesicular transport, organelles transport, and it kind of facilitates neural outgrowth, but it's mitigating the kind of functional consequences of tau loss. But what it's not doing is preventing tau hyperphosphorylation, and it's not preventing tangle formation. Okay, but it is allowing for that kind of neural outgrowth, and it's kind of compensating for that loss of tau. That's kind of the end of this kind of virtual session on Alzheimer's disease. I hope everyone found it helpful. And again, if you're kind of like a first year or second year student in life and sciences, whatever it is you're studying, please comment down below. I'd like to know, and I'd like to know how helpful you found this video. I tried to cover as much as I could, and I think a lot in exams and in essay style questions, a lot of the time you'll be asked to evaluate treatments in Alzheimer's disease. You'll be asked to and kind of link them back to that kind of that challenge in pathophysiology and why it's so problematic to treat. Okay, and kind of emphasize its degenerative nature, that kind of amplification loop. But yeah, I hope everyone found this helpful, and the next video I'll be uploading videos simultaneously on biochemistry. So we're going to continue with our kind of journey on like amino acid synthesis, protein synthesis, look at de novo synthesis of DNA. We'll go on to the glucose metabolism, and of course I'll be uploading 2 videos a week on pathophysiology. So we'll look at many inflammatory diseases like asthma, multiple sclerosis, arthritis. And yeah, I hope everyone found this session helpful.